Dear students, today in this lecture we will discuss sampling techniques part 4. The basic concepts about the sampling are the true value, accuracy and precision. True value is the value which is obtained from the population. Population which have all the observations, so the true values are the values which are based on all the observations present in the population. These values are exact values, accurate values. For example, the mean, the variance, standard deviation, all values are called true values. Accuracy. If we draw a small sample from the population and on the basis of the sample values we calculate the same measures, then the difference between the measures obtained from the sample and the two values is called the accuracy. Accuracy means how much sample statistic is closer to the population parameter. If the difference is less, then we have accurate measures. If there is more difference between the sample statistic and population parameter, then the values are called less accurate. Bayesian. If we draw more samples from the same population and on the basis of each sample we calculate the mean. If the mean of all the samples are closer to each other, then we will say there is more Bayesian. So Bayesian refers to the closeness of measures which are obtained from the repeated sampling. From this figure, we can understand the difference between accuracy and Bayesian. The left bottom figure have black dots which are obtained from the samples of the same population. We drew 8 samples from the population, and from those 8 samples we calculate the sample statistic. Those sample statistics are scattered in this figure in the form of black dots. And all these black dots are far away from the center. Our target is the central value. True value, the exact value, is in the center. And the difference between the calculated values from the sample and the true values is more. Therefore, there is less accuracy. And all these measures are far away from each other. Therefore, there is less Bayesian. Both accuracy and Bayesian are low in this figure. Now come to the bottom right figure. All the 8 results obtained from the samples are represented by the black dots and which are closer to the central value, the true value. So the difference between the true value and the sample statistics is less, which means the accuracy is high. But all these dots are far away from each other. So there is less Bayesian. Come to the top left figure. We will observe all the dots are closer to each other. This shows there is high Bayesian since all the values obtained from different samples, from repeated samples, those measures are closer to each other. So Bayesian is high, but still the measures is far away from the central value. Therefore, the accuracy is low and Bayesian is high. Now come to the in top right picture, all the sample statistics lies in the center of the figure and the true value is also here. So the difference between true value and the sample statistics are very low, therefore accuracy is high. As well as the values obtained from the sample are closer to each other as well, therefore Bayesian is high. Both Bayesian and accuracy are higher in this picture. Now we compare accuracy and Bayesian from one more figure. This is a dart game in which dart is thrown And this is the in the center of the figure. This is the target, and dart is closer to the target, then there is a high accuracy. Look at the three pictures. The leftmost have the red dot, which is the target, and all black crosses are the dots shots, and the shots are far away from the central value, therefore there is less accuracy. And the results are also not closer to each other, so there is less precision. In the middle of the figure, all shots are closer to each other, so precision is high, but still far away from the central value, therefore accuracy is low. In the right figure, we observe that all shots are in the center of all shots are closer to each other, therefore high precision, and all dots are in the center of the figure, which is the target value. Accuracy is high and precision is also high in this figure. We can see the results from here. Accurate but not precise, precise but not accurate, and accurate and precise both. This is the population in which capital N, mu, and sigma are the population parameters. N is the total number of sampling units in the population. Mu is the mean of the all observations present in the population. So this is the population parameter. Sigma is the standard deviation, which are based on the all observations in the population. The distribution obtained from this population is represented here with the blue line. This is the distribution of the population. In the center of the curve, there is mu, the population parameter, the average, and there are three samples, and corresponding means are x bar 1, x bar 2, and x bar 3. If we plot x bar 1, x bar 2, and x bar 3 on the same number line, then we will observe that x bar 1, x bar 2, and x bar 3 are scattered on the number line, and mu is in the center. The difference between x bar 1 with the mu, the difference between x bar 2 with the mu, and x bar 3 with the mu is called the sampling error. There are two types of the errors: sampling error. Sampling error are those errors which occur due to the process of sampling. Other one is non-sampling error, which occurs from the procedure of calculation and the measurements obtained from the sample. So total surface error is further divided into two categories: sampling error and non-sampling error. Generally, we have 5% errors, the sampling error, and 95% errors are the non-sampling errors, which are further divided into two types: non-response and responsive. Sampling errors is the error caused by observing a sample instead of the whole population. Whole population includes all sampling units, so the measures obtained from the whole populations are the true values, exact values. On the basis of small number of sampling units, the measures are obtained, and the sampling error is the difference between a sample statistic used to estimate a population parameter and the actual but unknown value of the parameter. Sampling errors are the errors of the representation. First of all, we take the target population from the population, and from the target population we construct a sampling frame. Sampling frame is basically list of the all sampling units. So there may be chance of error, and that error is called coverage error. Once sampling frame is formed, then the next process is sampling. A sample is drawn from the sampling frame, and the error occurs due to the sampling process is called the sampling error. Once sample has been obtained, then the respondents in the sample are willing to respond or not willing to respond. Non-response error may occur here. We can understand the sampling error from this figure. There are two curves. One curve is obtained from the population, and this is a distribution of population, and the curve has the mean in the center. So this is the line of the population mean mu, and the right side curve is obtained from the distribution of the sample. The sampling units, and its mean is x bar, so x bar lies in the center of this curve, and the difference is shown with the blue color. So that this difference is called the sampling error. In this figure, the rectangle is showing the population. Sample is a small part of this population, and outside this sample, this area is called the sampling error. Since our sample is small as compared to the population, the difference is more, the sampling error is high. In this figure, we can observe the sample is now more as compared to the previous one. The area outside this sample is now less. So the sampling error is less. We can understand that as the sample size increases, the sampling error decreases. If we increase the sample more than this one, then the sampling error will be decreasing more. Sampling versus non-sampling errors. Sampling errors which apply only the sample surveys are due to the fact that only a subset of the population is selected. And non-sampling errors which apply to all statistical processes is often categorized as coverage error, non-response errors, processing errors, and measurement errors. Non-sampling errors are due to the behavioral effects, due to the questionnaire or measurement error, or sometimes it is referred as poor sampling method. Non-sampling errors is also called as errors of measurement. First, we construct a questionnaire, and the questions related to our study are included in the questionnaire. And then questionnaire is used to obtain the data. So measurement is next, which are obtained from the questions we ask. There may be error, and that error is called measurement error. And once we get the observations from the respondents, the responses are written in the form of document, or we can enter the data into the Excel sheet in the computer. Then there may be the error, which error is called the processing error. So all these errors are called as non-sampling errors. Sampling errors and sample size. As we increase the sample size, the distribution of sampling error is in the form of bell shape, and we are measuring sampling error here. In the center of there is the mean, and the tails are spreaded. The tails are far away from the central value mean. So there is more sampling error. In the next figure, sample size is double as compared to the previous one. The tails are now squeezed in this figure, and peak is more high. In the next figure, we can observe the sample size is now more, and all values are now scattered around the central value mean. In next figure, if we increase the sample size, then more observations lie in the center, and that's why peak of this curve is higher, and less values occur at the tails. Whenever sample size is increased up to the capital N, which is the population size, it means if we select the all population as a sample, then population mean mu and sample mean x bar both are same. So all of the observations lies At the same point, then sampling error is now 0, and the figure obtained in the form of a single line, which is the line of the mean. There are one more type of the errors, that is called the bias. The errors which makes up all flaws in the study's results. You can observe from this figure, there are 3 persons on the one side and 1 person is on the other side. Although 3 persons weight is more as compared to the weight of 1 person, but still 3 persons are lighter as compared to the 1 person. So this is due to the bias. Bias refers only the error that is systematic in nature. Research is biased when it is gathered in a way that makes the data's value systematically different from the true value of the population of interest. Different types of biases. Researcher bias. His own bias may be a cause of the error. Survey bias, respondents bias, and non-response bias. These are the different types of biases. Whether it is in the selection process, the way of questions are written, or the respondent's desire to answer in a certain way, bias can be found in the almost all of the surveys. There is an example of the bias. For example, including a question like, do you drive recklessly in the public safety survey would create systematic error and therefore be biased. The reason it is considered systematic is that many respondents would answer the question falsely in one direction and selecting no, even they are a bad driver. If you are asking a question like this in the public place, definitely you will get the wrong answers from the respondents, and respondent will be saying that we are a good driver, although they are actually not. First figure is showing the target population, which is this one. From this target population, the difference x bar t minus x bar is called the total error. From this distribution, we take a survey population, that is called the target population. And from this target population, one more mean is calculated, that is called x bar s, survey population. And the difference between the true value and the survey mean is called the non-sampling bias. And then we draw a sample, and this is called the sampling distribution, and its mean is called the expected value of x bar. So the difference x bar, expected value of x bar minus x bar, is called the sampling variability. And actual one sample is drawn here. This is one sample, and its mean is in the center x bar here. And you can observe there are distribution of, this is the small error, which is due to this one. Next error is the sampling bias, and next error is the difference between sampling variability. So the total difference is from here to here is called the total error. Thank you so much for watching this video. Allah Hafiz.